again, this picture, to me, looks like a nice histological picture of what's this called?
Soma?
Oh, that's this. The whole thing is a, yeah, giant multipolar nerve, right? You were looking at that under the microscope yesterday. Okay, so let's start with terminology, okay? So when we're talking about the nervous system, we have to get the basic definitions down, okay? We've already talked a little bit about neurons. So when we were talking about muscle and the neuromuscular junction, I briefly mentioned the anatomy of a neuron. So we'll kind of expand on that, add a little bit more detail to that. But the basics we've already generally covered. Okay, so a neuron is the same thing as a nerve cell. Okay, so a nerve cell and a neuron are the same thing. Now, the neuron that we're used to, I have to sneeze. Okay, there we go. So the neuron that we're familiar with, okay, is the one that has a cell body, comes down into an axon hillock, right? You've got one ending where you've got those axon terminals, the synaptic knobs, and then you've got little, what are the strings that are coming out the other end called? Dendrites, right? Yeah, and then in the middle you've got a cell body with a little dot, which is the soma, which is where all the DNA is. And then the region of the neuron where the axon hillock is where the electrical impulse is generated. So, okay, so that's like the basic neuron anatomy that we talked about in muscle unit. So now, a neuron is a nerve cell, okay? But that's not the same as a nerve, okay? So to define a nerve is going to be a collection of axons in the PNS. And PNS stands for peripheral nervous system, which we'll define in a little bit, okay? So a nerve is a collection of axons in the PNS. A nerve is not the same as a nerve cell. It's not the same as a neuron. Nerves are axons only. Okay? If we mention the term fiber, okay, so if we're talking about fiber, now in the muscle unit, a muscle fiber was the same thing as a muscle cell, right? In the nervous system, a fiber is describing an axon only, okay? So we have to remember that. So a fiber is an axon in the nervous system. Now, ganglion is singular, ganglia is plural, okay? A ganglion is defined as a collection of cell bodies in the PNS. Okay, so that's what a ganglion is. It's a collection of cell bodies in the PNS. PNS being peripheral nervous system. Okay. All right, we've talked about synapses. How can you, like, just in general, just kind of simply define a synapse? How would you define a synapse?
The cleft between two axons.
Say that one more time.
The cleft between two axons.
Okay, so that would be the synaptic cleft. So the synapse is in total the connection, right, between two cells, whether it's a muscle and a nerve or a nerve and a nerve. Okay, so kind of what happens at a synapse? What are the two cells doing?
Communicating.
They're communicating, okay? So you can say the synapse is a location of communication, right? This is where two cells are going to be talking to each other. So we were talking about the neuromuscular junction, but now we're going to talk about the communication between a nerve cell and another nerve cell in the nervous system. Sorry, my writing is terrible. Okay. All right, so receptors, we've talked about receptors. And receptors in the body, we have different kinds of receptors, right? So we've got chemoreceptors, baroreceptors, mechanoreceptors. How would you describe just a general receptor? How would you define just a general receptor?
Responds to signals.
So receptors are going to be sensitive to some sort of stimulus, right? So mechanoreceptors are going to be sensitive to mechanical change. Baroreceptors are going to be sensitive to changes in blood pressure. So receptors are going to be sensitive to some sort of stimulus, okay, depending on what kind of receptor they are. Okay, now effectors, we haven't really heard of this term yet, but effector is essentially defined as what is stimulated by a nerve. And in our story that we're going to talk about today, it could be muscle tissue or it can be glands. And when I mean muscle tissue, it could be cardiac, skeletal, or smooth. Okay. So an effector is what is being stimulated by a nerve, or what is being stimulated by a nerve. Okay, electrochemical communication, we know this already, okay? We know at a synapse we have a presynaptic cell, we have a postsynaptic cell. In order for them to communicate, the electricity coming down the presynaptic cell is turned into a chemical signal, right? You're using neurotransmitters, the one that we talked about. What neurotransmitter did we talk about?
Acetylcholine.
For muscle contraction is acetylcholine. Yeah, there are other neurotransmitters out there, serotonin, epinephrine, norepinephrine, depending on where you're talking about. And then that neurotransmitter transmits that electrical signal from the presynaptic cell into the postsynaptic cell by stimulating that postsynaptic cell membrane. Okay, so that's electrochemical communication. And then we know action potentials. You either get an electrical impulse or you don't. And if you get one at the axon hillock, if it's generated, it goes all the way down the length of the axon to the axon terminals. Okay? There's no halfway, there's no partial. It's an all or none event. Okay? Okay. And neurons are unique cells in that they can generate their own action potential. They don't need to be stimulated. They're not like muscle tissue. Muscles need nerves in order to be excited. Nerves can generate their own impulses when they're communicating with each other. Okay. Now, another term that I want to make sure I'm communicating in this list is nuclei. When we talk about nuclei in the nervous system, it's going to be a collection of cell bodies in the CNS. And that's central nervous system. Again, we're going to define CNS and PNS here shortly. Okay, and then the last term that we need to talk about, which I briefly mentioned yesterday, is a tract. And a tract is a collection of axons in the CNS. Okay? So those pyramidal cells yesterday that you guys were looking at, they belong in tracts. So the axons of those cells are found in pyramidal tracts. Okay, and we'll talk more about that later. All right. Okay, questions about definitions. Again, we'll repeat ourselves here as I go along. Okay, so let's define what is CNS and what is PNS, okay? So when we're talking about the central nervous system, okay, central being the key word, okay, we're talking mainly about the brain and the spinal cord. That's considered the central nervous system, okay? Anything coming outwards of the spinal cord, okay, is considered peripheral nervous system. So all the nerves that we have in our body, the peripheral nerves that you guys are gonna look at today in lab, femoral, brachial, axillary, okay, tibial. Those are all peripheral nerves, part of the peripheral nervous system. And associated with those nerves are ganglia, okay? So how did we define a nerve? A nerve is a collection of what? Axons in the PNS, right? Very helpful when you repeat these things over and over again. And a ganglia, or ganglion, which is singular, is a collection of cell bodies in the PNS. So there you go. All right. Okay. Okay, so with structure comes function, all right? So what is each part of the nervous system responsible for, okay? So again, central nervous system, we're talking about brain and spinal cord, all right? So on this picture, the yellow arrow is what's signifying CNS, okay? The blue and the red here, those arrows are signifying PNS, okay? So functionally, the central nervous system, being the brain and the spinal cord, is responsible for analysis. It's responsible for taking in information, analyzing what it is, and generating some sort of a response, okay? The peripheral nervous system, okay, is designed to pick up the stimulus, whatever it is, okay? And since we're talking about sensory stimulus, sensory stimuli, this could be, the example here is vision, so it could be light waves, it could be sound waves, it could be touch, light touch, deep pressure, tickle, okay? Temperature, it could be pain, okay, any kind of a sensory stimulus. Those kind of stimuli are gonna be picked up by receptors that are sensitive to that particular stimulus, and it's gonna carry that information from the periphery towards the CNS. And we've used this term afferent already, right? And so afferent means leading towards, same thing, all right? So sensory information is coming in from the periphery, headed towards the CNS. So it's called afferent information, okay? And since it's coming from the periphery, it's part of the peripheral nervous system. So the peripheral nervous system is designed to bring in sensory information to the central nervous system, the brain and the spinal cord, for it to be analyzed. What is it that you're looking at? What is it that you're hearing? What type of pressure are you feeling? Is it a tickle? Is it a scratch? Is it pain? Is it pressure? What kind of temperature? Is it hot? Is it cold? Okay, so the central nervous system is designed to analyze whatever it is that's coming in, and if necessary, generate some sort of response. And that response is then converted into a motor or efferent signal. Efferent meaning what? What does efferent mean? Leading away, right? E for exit. Yep. So the central nervous system, after integration and analysis, it's going to generate a motor response, and that motor response is going to hit an effector. Okay, and an effector in our story is going to be muscles or glands. And the overall effect, if it was muscle, for example, in this situation, what would the effect be?
Movement.
Right, or contraction. And if it were a gland, what would happen?
Releasing.
Secretion.
Secretion, right? The gland would secrete something, yeah. Yep. Okay. All right, so those are the functional divisions, right? So the PNS is sensory and motor information. The CNS is integration and analysis. Okay. Structurally, the PNS involves nerves and ganglia. The CNS structurally involves what?
Nuclei.
the brain, and the spinal cord. Okay. Again, just laying like a foundation here of terms. Okay. All right, so let's expand upon the functional divisions because we can further break down sensory and motor information depending on where it's coming from in the body, okay, where it's going to in the body. So we know at the top, central nervous system is the analysis center. So the blue arrow heading towards the CNS from the PNS, that's going to be sensory information. The red is going to be motor information, okay? So if I had to add arrows to our diagram, motor is heading outward and sensory is heading inward, right? Okay, so functionally we can break down the peripheral nervous system into sensory and motor divisions. Okay, sensory bringing information towards the CNS and motor bringing information away from the CNS, okay? So, we can break down sensory information even further into somatic sensory information and visceral sensory information. Same thing with motor information. We can break that down into somatic motor information and visceral motor information, okay? Depending on where those effectors are in the body. Sensory, depending on where that stimulus is being provided in the body, okay? So, let's define the term somatic. So the best definition for somatic, in terms of understanding, is a body region that's not in a body cavity. So, 3 perfect examples of somatic areas of the body include your skin, your bones, and your skeletal muscle. Okay, so those are the 3 somatic areas of the body. Okay, those are areas of the body that are not located in a body cavity. Okay, you can't find skin in a ventral body cavity or a dorsal body cavity. It's all over the body. Same thing with bones. Same thing with skeletal muscle. Okay. All right, so if we're talking about somatic sensory information, okay, this is going to be sensory information that's coming in from the skin, your bones, or your skeletal muscle. And that could be sensory information involving pain, pressure, touch, tickle, temperature, anything sensory, vibration, okay, stretch. Okay. Visceral sensory information, what does visceral mean? Of or pertaining to organs, right? So this is deeper sensory information. This is sensory information coming in from your inner organs. Same type of sensory information. Stretch, temperature, touch, pain. It's the same kind of information, just coming from a different part of the body, okay? Now, one thing we have to keep in mind is that we have special senses. And hearing, vision, which we're going to look at tomorrow in lab, and our sense of equilibrium is considered somatic senses. Okay? Taste and smell are considered visceral sensory information. And the way I remember that is that taste and smell, when I think of that, I think of like digestion, I think of like digesting something you just ate that was really good. And so that's coming from your viscera, that's coming from your organs. Your stomach is telling you that it's happy, it likes what it received. So the special senses of taste and smell are considered visceral sensory. Okay, so now for the motor division, which is outgoing information to effectors, right? And effectors are what is stimulated by a nerve. We have 2 choices. We have either somatic motor or visceral motor information. So somatic motor information, okay, involves effectors that are only skeletal muscle. So somatic motor neurons are only going to innervate skeletal muscle. That is their only effector. Okay? And because we have voluntary control over our skeletal muscles, this is the voluntary motor pathway. Okay? Somatic motor. Okay? So visceral motor then, same thing, the effector is going to be some sort of muscle tissue or glands, but for visceral motor, we don't have control over that. We don't have control over contraction of our inner organs, okay? So those effectors are going to include cardiac muscle, smooth muscle, and glands. Effectors that we don't have control over, involuntary, okay? So now, for this unit of study, okay, the lecture exam next week, all we're learning is terminology, central nervous system, and peripheral nervous system. We've talked about the autonomic nervous system, okay, we've talked about sympathetic and parasympathetic control in the body, okay, but we're gonna talk about the autonomic nervous system next unit, all right? But what I want you to know is the visceral motor division of the PNS is considered the autonomic nervous system. Okay? So the autonomic nervous system is considered the visceral motor division of the PNS. And those effectors are going to include cardiac muscle, smooth muscle, and glands. Okay, questions about any of this? Okay, so this picture essentially is just for your reference. It's giving you examples of sensory information and motor information. So for example, skin. Sensory information coming in from the skin is coming in on a somatic sensory fiber, okay, which is an axon. A very long axon. The stomach, if it's giving you sensory information that it's stretching, it's going to give you visceral sensory information. If you've got a skeletal muscle that's contracting, then you know that it's going to have a somatic motor fiber going to it that's causing that contraction. Okay? And a visceral motor fiber, because it's considered autonomic nervous system, if it's stimulatory, okay, it could be potentially sympathetic. If it's inhibitory or happening when you're resting and digesting, like urinating, then you're going to have a visceral motor fiber going to that particular effector. So again, cardiac muscle of the heart and smooth muscle of the bladder. Okay, so those are just nice examples to kind of summarize what it is we just talked about. So now, let's practice. So we have 4 choices, okay? We have 2 sensory choices and we have 2 motor choices. So, for example, this pathway carries information from the skin to the brain. What option is it?
Somatic.
Somatic, sensory, right? Taking information from the skin to the brain. So you know it's sensory, it's going to the central nervous system, and it's somatic, okay, coming from the skin, okay? What about this kind of neuron would stimulate skeletal muscle? Somatic, motor, right? It's only going to skeletal muscle, right? And it's somatic because it's addressing skeletal muscle, which is one of the 3 somatic areas of the body. Okay, good. What about information about your stomach feeling hungry?
Visceral.
Visceral, sensory, right? Yep, good. Okay. Fantastic. Getting it. All right, good. Okay, nothing really new here. We already know, okay, that neurons can generate their own electricity, all right? They can conduct that electricity all the way down the length of the axon using saltatory convection. We talked about that. And then at the axon terminals, terminal boutons, you're gonna release a neurotransmitter, depending on where we're talking about in the body. Acetylcholine is one we've talked about. And that results in an electrochemical response to some sort of a stimulus. Nothing new here. It's just information you guys already know. So let's break down neurons into functional classes, okay? Then we'll talk about structure. All right, so functional classes of neurons, we have three. We have sensory, we have motor, and we have interneurons, okay? Sensory, we know, can also be called afferent, heading towards the central nervous system. Motor is also considered efferent, heading away from the central nervous system towards some sort of effector, okay? So let's start with sensory. Sensory neurons are going to bring in information from the periphery, and they almost always end at the brain and spinal cord. Pretty much always. It almost always just leaves always the window open, but always. In the brain and spinal cord, okay? So they're bringing information in from the periphery, from the PNS to the CNS for analysis. Okay, stretch, temperature, pain, vibration, okay. Motor at the bottom. So motor neurons are gonna end on some sort of effector. So they're gonna terminate on some sort of effector. And depending on whether it's somatic motor or visceral motor, okay, then you know what your effector is. If it's somatic motor, what's the effector?
Muscle?
So what kind of muscle?
Skeletal.
Skeletal muscle, right? And if it's visceral motor, what are your choices?
Smooth muscle. Cardiac.
Smooth muscle, cardiac muscle, or glands, okay? Okay. So that leaves us with the last example in the middle, which is interneurons. 99% of all your neurons are interneurons. Okay? And they are designed to analyze. So interneurons are always found in the CNS. Okay? Sensory and motor neurons are found in the PNS. Interneurons are found in the CNS. And a majority of them are interneurons, which gives you how complex our nervous system is. Because you have all kinds of stimuli coming in from all parts of the body, and it's the nervous system's job to analyze that information and determine whether or not a response is necessary and where in the body. Okay, so we'll come back to this picture a lot because it's just a great example of neurons and synapses and connections. So what this is essentially just showing you, okay, we're gonna look at this in lab today, nicely. So you're looking at a transverse section through the spinal cord, okay? And on either side of the spinal cord you have two roots. And these roots ultimately connect to what's called a spinal nerve. Again, you're gonna identify all this in lab today. So spinal nerves go out into the periphery and become peripheral nerves, like your femoral nerve, your sciatic nerve, your brachial nerve, your radial and ulnar nerve. All of those nerves emanate from some sort of spinal nerve that originates somewhere on the spinal cord, okay? All right, so this example, this pathway example, is just showing you information that's coming in from the skin, okay? Looks like it's coming in from the dermis. So you got receptive endings that are picking up some sort of stimulus. Okay, maybe it's stretch. Okay? Say for example, you're at the gym and you're stretching your gastrocnemius. Okay? Getting stretched, ready to work out. So that sensory information is coming in from the periphery to the CNS, okay, on an afferent neuron, on a sensory neuron. Okay? Now, because we're talking about skin here, what kind of sensory neuron? Somatic or visceral? Somatic. Somatic sensory. So that information's coming in, and it's synapsing, oh, what a big surprise, in the CNS with an interneuron. Okay, 99% of all neurons. So those interneurons are picking up that information and they're analyzing it. What is it? Oh, it's stretch. Oh, it's coming from the skin. Okay? Because you happen to be stretching your gastrocnemius muscle at the gym. And you can feel the muscle, but you must be really stretching it good because now you're getting to the point where you're stretching your skin. And your skin is getting fired off and sending information to your nervous system, which is being analyzed. And that information could be stretched. It could be stretched to the point of pain, perhaps. Okay? So that interneuron then is going to communicate with a motor neuron. And that motor neuron is going to leave efferently. It's going to leave the CNS via the PNS out of spinal nerve. And it's going to go to an effector. And this effector here looks to me like skeletal muscle. I see striations. So what kind of motor neuron is this? Somatic motor, right? Okay. And it's going to terminate on, for example, your gastrocnemius muscle. And it may tell your gastrocnemius muscle, hey, you need to contract. Because you are stretching it just way too much and you need to stop. So your response is going to be like, contract the gastroc, ease off the skin. All right, it's a protective mechanism. Okay. Okay, so all of that has to be analyzed and processed for a response to happen. Okay, lightning fast, obviously. Lightning fast. You blink and it's done. It's done even before you blink. Okay. Okay. Now, other things to think about here. A ganglion. That's an enlargement here. And we defined a ganglion as a collection of what? Cell bodies where? In the PNS. There's a cell body there. Okay. We're in the PNS because we're out of the spinal cord. So that is a ganglion. It's an enlargement that is a collection of cell bodies in the PNS. Okay. How did we define a nerve? A nerve is a collection of axons in the PNS, right? We're in the periphery. What's in a spinal nerve? It's only the axons of these neurons. And we'll talk about structure here in a moment. Okay? Okay. I'll come back to this picture. Okay. So, the basic functional unit of the nervous system is the neuron. Okay? Basic functional unit of the muscle system was what? The sarcomere, right? Yeah. Every system has one. Okay. So this is just your typical beautiful neuron that's depicted in most textbooks, including yours, all right, that we talked about. So this is your standard neuron anatomy, cell body, hillock, axon. We've got Schwann cells around it to insulate that electrical activity, make sure it doesn't dissipate, okay? Dendrites receiving information, okay? And then the axon terminals with those terminal boutons, and that's what's holding the neurotransmitter. Again, with structure comes function, right? We're like, this just a repeating concept over and over again in anatomy. The synapse, again, nothing new here. Okay, we've seen this picture. We know we have vesicles. We know there's mitochondria to drive, okay, the exocytosis. Now we're just talking about nerve-to-nerve communication. So again, nerves similarly come really, really, really close to each other, but do they touch? No, they don't. So that's why we need electrochemical communication. Okay, so it's the same thing. Synapse and all. Okay. Again, this is just repeating what you guys already know. So you know that neurotransmitters are going to be released. They're going to bind to specific receptors that are sensitive to them, and then they're going to stimulate that postsynaptic membrane and generate an electrical response in the postsynaptic cell. Nothing new. Okay. Okay. So now let's talk about the structural classes. So the structural anatomy of neurons, we have three options, okay? The first one is multipolar. Multipolar is your classic neuron. That's what we looked at under the microscope yesterday, the giant multipolar neuron, okay? Multipolar neurons, okay, the most common in the central nervous system, okay? CNS includes what two structures?
Brain, spinal.
Brain and spinal cord. Okay, that's where you're gonna find multipolar neurons. Okay, they have 2 or more dendrites, and those neurons are either gonna be motor in function, or they're going to be interneurons. So you have 2 options, and they're gonna take on this anatomy. Okay? Again, if interneurons are 99% of the neurons that you find in the CNS, then most of them are gonna be multipolar. Okay? All right, we'll get back to bipolar in a minute. Let's go to unipolar. Okay, so unipolar neuron is a little bit different in anatomy. All right? Unipolar, I think of one pole, one single pole coming off of the cell body, and then it splits. This whole thing is an axon, but it splits into 2 different what they call processes. Okay? The peripheral process goes to the periphery, okay? Picks up information from the periphery. It's got receptive endings, okay, that are sensitive to stimuli. And then you have a central process. The central process is gonna bring that information to the CNS. That's why it's central, okay? So all unipolar neurons functionally are sensory, all of them. Don't worry about pseudo-unipolar, it's just another term we don't need to worry about. Okay? So now let's go back one here to bipolar. Bipolar is special because you have a cell body, and coming off of it is 1 dendrite and 1 axon. Okay? You only find this in certain parts of the body. It's always sensory. You're only going to find it olfactory, eye, and ear. Okay? So smell, sight, and hearing. Sensory only, bipolar. So the only parts of the body you'll find bipolar. Okay? Okay, and we'll talk more about bipolar when we talk about special senses, when we get to that in lecture, but for now, you just know it's one of the 3 structural classes. It's sensory, it's only found in 3 parts of the body. Okay, so if we go back to this picture that I like so much, the blue neuron here, okay, we said functionally, what kind of information is being, what kind of information is traveling through this neuron? Sensory or motor? Sensory, okay? Okay, so sensory. Structurally, you have 2 options. You have either bipolar or you have what? Unipolar. And what does that neuron look like? Unipolar, right? So we're talking about skin, okay? Anywhere other than olfactory, eye, ear. So that is structurally unipolar. You have your peripheral process, and here's your central process, bringing information in. Okay? So now, interneuron here, and red motor neuron, structurally, they look the same. And what did we say structurally interneurons and motor neurons are?
Multipolar.
Multipolar. So you see that they are multipolar in the picture. Okay. Okay. All right. Okay, so aside from neurons, we can't ignore supportive cells. And you've looked at some of these under the microscope. You looked at astrocytes yesterday, okay? So for supportive cells of the CNS, central nervous system, we have 4 that I want you to know, okay? So astrocytes help with, glutamate is a specific kind of neurotransmitter, just remember that they help with neurotransmitter uptake from synapses, okay? Because we know that after an electrical impulse passes, that neurotransmitter has to be taken out of the synaptic cleft in order for the stimulus to stop, right? Because we're not constantly stimulating. Stimuli come and go. So astrocytes, okay, look like spiders. They have multiple extensions. They can affect multiple neurons. Okay, so those are supportive cells. Microglia or microglia are basically just the cleaner uppers, okay? So they pretty much engulf anything that's pathological and get rid of it, okay? They have a lot of extensions too, so they can affect multiple neurons. So they're roaming around in the CNS. Then you have what are called ependymal cells. And ependymal cells are unique in that they have cilia on their apical surface, okay? And they line all the fluid-filled cavities in the CNS, okay? So the lateral ventricles. What other ventricles did we look at yesterday?
Third and fourth.
The third and fourth ventricles. The cerebral aqueduct. The central canal of the spinal cord, which we'll look at today. Okay? All of those fluid-filled areas are lined with ependymal cells. And ependymal cells are designed to circulate CSF, which is cerebrospinal fluid, okay, and move it along. Okay, it doesn't make it, but it circulates it, keeps it moving. And that fluid is designed for nutrients, waste removal, okay? All right, in the CNS we have the last cell, which is called an oligodendrocyte. An oligodendrocyte is kind of like a Schwann cell, but it's in the CNS. So the oligodendrocytes make myelin in the CNS, okay? So oligodendrocytes have multiple extensions that come off of them, and they can create myelin on multiple axons of multiple neurons, okay, so they can multitask. So those are supportive cells of the CNS, there's 4. Now for the PNS, there's only 2. The first one is called satellite cell. And the satellite cells are designed just for those sensory neurons, those unipolar ones, is they just surround the cell body and protect it. Okay, so they just add a layer of protection to the cell bodies of those unipolar sensory neurons in the PNS. The Schwann cells, which we talked about before, are the cells in the PNS that make the myelin, okay? Now, Schwann cells are different than oligodendrocytes in that each individual Schwann cell wraps itself multiple layers around a portion of the axon, okay? So you're gonna have multiple Schwann cells along an axon, versus oligodendrocytes can create myelin on multiple neurons. They just coat the entire axon. Okay, so a different mechanism of creating myelin from each of those 2 different kinds of cells. So again, this is just emphasizing what I just said. So oligodendrocytes, you're gonna have multiple extensions. They're gonna create nice big sheaths, big blankets, okay, around the axons. And then in the PNS, each individual Schwann cell carries multiple layers in and of itself. So you're gonna have multiple Schwann cells per neuron, per axon, I should say, because that's what they're lining. Now, there are unmyelinated axons. Not all axons are myelinated, okay? It doesn't need to be that way, okay? So Schwann cells can still provide structural stability to unmyelinated axons. Okay, so Schwann cells can cover multiple unmyelinated axons and just provide structural stability, okay, in the PNS. Okay. That's that's all I want you to just remember. Not all axons are myelinated. Okay. All right, so we come back to the same picture again. So, when we're talking about information, okay, that's coming in from the periphery, from the PNS, okay? The peripheral nervous system includes two structures. What's one of them?
Spinal nerve.
Okay, a nerve, nerves. And collections of cell bodies called ganglia. Okay, so those are PNS-related structures, okay? So we know functionally, okay, the peripheral nervous system is going to provide either sensory information coming in or motor information going out, okay? What else did I want to say here? Interneurons, we know, are 99% of the neurons in the body. Now, one thing that, again, you're going to identify this in lab today, which is nice, is we're going to identify gray matter versus white matter in the central nervous system. Okay, that's brain or spinal cord. We'll talk more about the brain when we talk about CNS, okay? So gray matter contains different structures than white matter. Okay, white matter is called white because it contains a majority of a certain protein. Anyone have an idea of what that is? That insulates electricity on the axons? Myelin. Okay. So white matter is composed of myelinated, okay, axons. There's some non-myelinated, yes, okay, but a majority are myelinated, and that's what gives white matter that color, okay, lighter color. Now, we've defined tracts already. So tracts are what now? Collections of axons in the CNS. Okay, so now it's making a little bit more sense. Okay, so tracts are going to be lined with myelinated axons, not all, but a majority of them, in the CNS, because we're talking about brain and spinal cord. Okay, so that's what you find in white matter. A bunch of other structures we find in gray matter. And in gray matter, in the spinal cord, it's kind of nice, because gray matter kind of takes on this shape of like a butterfly, okay? So the white matter is kind of in the periphery, and then deep to the white matter is the gray matter in the spinal cord, okay? So in gray matter, let's just look structurally at what we find in it. So let's make a list. Give me some structures that we find in gray matter.
Synapses.
Okay, synapses, we can get even more specific than that. So what parts of the synapse? Give me anatomical structures.
The dendrites,
Okay, we can find dendrites.
the body,
You can find cell bodies of what? Interneurons and motor neurons. Okay, so in gray matter, you can find dendrites. You can find cell bodies. And we said specifically of interneurons and motor neurons. Okay, we can find synapses. Okay. And what are these here that I'm circling? What are those? Axon terminals. We can find axon terminals, because if we can find dendrites, we should be able to find axon terminals. Okay. In gray matter as a whole, you can find interneurons. entire interneuron can be found in the gray matter. Okay, you can find the cell bodies of motor neurons, and you can find the axon terminals of sensory neurons. You can find the entire interneuron in the gray matter. So not all interneurons are myelinated, okay? Okay, because gray matter is gray because there's no myelin, okay? So myelin is used to insulate a signal and increase the speed, all right? So, for example, if I were to take this picture, and we'll talk more about this later on, but just in general, if I were to create another connection, okay, of say another interneuron that was maybe synapsing on this interneuron and heading upwards, say towards the brain, that interneuron is probably going to be myelinated and found in the white matter, because that's going to allow for increased speed, okay, and the ability for that interneuron to synapse somewhere higher for conscious level processing, right, to give you the conscious perception of what it is that you're sensing. Am I sensing tickle? Am I sensing temperature? Okay, is my hand on a hot stove? Am I overstretching my gastrocnemius in the gym and pissing off, you know, my muscle and my skin? Okay, so depending on what's needed of that particular neuron is where you're gonna find the myelin, is what I mean for this, without getting too in-depth. Okay, so based on all these structures that we find in gray matter, what do you think the function of gray matter is in general? We've got synapses, we've got dendrites, we've got axon terminals, we have interneurons. What's the general function of gray matter?
Site of communication.
Okay, communication, but even more, what else? Analysis, right, and integration. So gray matter is involved in analysis, okay? It's part of the CNS's job to analyze and integrate the information. And you've got all those structures there that are gonna promote that. Promoting synaptic communication, promoting analysis. Interneurons receiving the information, analyzing it, generating a response, communicating with the motor neuron, okay? That's the function of gray matter. So now white matter, where we find mainly myelinated axons, okay? What would we say in general the function of white matter would be? Yeah, you could say like transport, right? Like transporting that signal very, very quickly, up or down the chain. Okay. All right. Okay. Now, another point, and I'll repeat this again, but since we're looking at this again, we know that nerves are collections of axons, okay, in the peripheral nervous system, okay? What kind of information do nerves contain? Functionally. What's the red and the blue mean? Sensory and motor, right? So nerves, all nerves of the body contain both sensory and motor information. And I'll repeat that again in lab. Okay. So what happens is basically, depending on whether it's sensory or motor information, it gets routed to a different part of the CNS, okay? And we'll talk more about that in a bit. So when you guys get into lab and we look at the spinal cord model, it's gonna look just like this, okay? The gray matter, again, looks like a butterfly. And you'll notice that posteriorly, so this is the back side, this is anterior, okay? Posteriorly, the gray matter, you don't really see it on this picture, but it extends all the way to the end of the cord. Okay, so technically, so imagine this is all colored in. So the gray matter extends all the way to the end, okay, on the posterior aspect of the spinal cord, all right? On the anterior aspect, it does not, okay, but posteriorly it does. And this is helpful because when you're looking at the model, you gotta know whether you're looking at the back side or the front side of the spinal cord in order to identify proper structures. Like we're gonna identify horns, we're gonna identify columns, central canal right down the middle where you've got cerebrospinal fluid flowing through. And the reason why the posterior horns of gray matter touch the outside is because they are collecting sensory information coming in from the periphery. So again, with structure comes function. Okay, so sensory information is coming in, and it's coming into the gray matter where those sensory axon terminals are located. Okay, so that's why the gray matter posteriorly makes a connection to the outside of the spinal cord, to the actual borders, because it's receiving information. Okay, motor does not. Motor information just leaves the gray matter for a hot second, goes through white matter, and then immediately enters a root, which is going to connect to a spinal nerve. And we'll talk about roots in a bit. Okay. Repetition is key. Nerves. Collections of axons in the PNS. This looks a lot like a muscle cell. Okay, straws upon straws upon straws. You've got 7-11 straws, you've got your traditional straw, and then you got your boba straw. So your boba straw is wrapped around a nerve in its entirety. And epineurium is the outer lining. Okay, just like epimysium was the outer lining of a muscle cell. Collections, okay, of 7-11 straws, which we call traditional straws, are fascicles. Fascicles have perineurium. And then you've got your 7-11 straws, which are individual axons, okay? Those are endoneurium. And you guys identified endoneurium on the neuron model. And we've got blood vessels in here. Okay, so our nerves, organs,
Yes.
yep, got different tissue types, right? All performing a common function. So nerves are organs. All right. All right, let's skip reflex. Yeah, let's just go back here for a moment. I just want to define something because you're gonna learn it in lab, so let's just write it on here. That way it just helps, the repetition is just helpful. Okay, so let's label these two areas. We have a posterior root, and in the front here, we have an anterior root. Okay? That's root, not roof. So, roots allow a nerve to connect to the CNS, and in this example, to the spinal cord, okay? So roots allow a spinal nerve to connect to the CNS, in this example, the spinal cord, okay? Because the posterior root only contains what kind of information? Sensory. And the anterior root only contains what kind of information? Motor, right? Sensory is going in. Motor is coming out. All nerves carry both kinds of information, but once you hit the roots, that information splits, okay? Because one's inbound, the other one is outbound, all right? So you have to have those roots in order to direct traffic properly. Okay, reflexes, we're gonna get back to, okay, because I want you guys to like simmer on this information first, then it'll make a lot more sense when we talk about the two different kinds of reflexes. And it's just a practice of the terminology that we just talked about, so we'll get to reflexes. Okay, as far as nerve regeneration goes, it's kind of like fracture healing. All I want you to know are the few steps, okay? So if a nerve, for example, is broken or damaged, so this is just showing you like an axon that's been destroyed, okay? You've got Schwann cells that also have been a little bit of destruction around them. So what happens is, first the macrophages come in, okay? Macrophages used to be monocytes. Monocytes get the sense that there's an injury, they leave circulation, they turn into macrophages, and they turn into the garbage trucks. So they get rid of all the axon debris, they get rid of the Schwann cell mess, okay? And then the Schwann cells start to lay down more myelin. So as they're laying down more myelin, they form kind of what's called a regeneration tube, and around that myelin tube, the axon starts to sprout. So it starts to sprout different branches, and then ultimately those branches will solidify and come together. And this takes years. This is like slow going, all right? If full regeneration happens at all. Just because a nerve regenerates doesn't mean it's going to regenerate its function. It may regenerate its structure, but it may not be perfect and function may be lost, right? Okay? So then you've got essentially the Schwann cells kind of backing off, doing their usual thing, and you've got that axon back together. So that's nerve regeneration. Again, a source of lots of research. You know, there's lots of accessory devices that, you know, paralyzed people use to help assist them. You know, paralysis is not reversible, unfortunately. So there are certain types of nerve injuries where you can get full recovery if you're lucky, you know. But it takes a long, long time, and it just depends on what your body is willing to give you. So, all right, so that's it for nerve regeneration, just the 4 steps, that's it, okay? And it takes a long, long time. Okay, so this is just, we're just kind of tying things together. So you don't have to memorize this picture. I'm not gonna put this picture on the lecture exam. I just want you to kind of get the general gist of what it is we're talking about. So, down here looks familiar, all right? So this is just showing you a basic pathway and the communication that occurs between the peripheral nervous system and the central nervous system, okay? So our example here, if someone's driving a nail through your shoulder, what kind of stimulus would that be? Pain. Okay, yeah. So we've got a painful stimulus, okay? And it's firing off those receptive endings on the peripheral process of structurally what kind of neuron?
Unipolar.
Unipolar. Functionally, it is sensory. Okay, remember, get structure versus function. Okay. All right, so we got unipolar. So we got the peripheral process taking information in. You got the central process taking information through the posterior root. It's going through the gray matter and it's synapsing with, what a big surprise, in the gray matter and interneuron. Okay? Interneurons are structurally multipolar. Functionally, they're basically just analyzers. That wasn't a trick question. Okay. So they're the analysis, all right? They're going to synapse on a motor neuron, which is going to take information out the anterior root through a spinal nerve to an effector. And since this effector looks like potentially what muscle?
Skeletal.
Looks like your deltoid, so your skeletal muscle. So what kind of motor neuron is this?
Somatic.
Somatic motor. Yep. Structurally, what is it? Multipolar. Yeah. And here's another, what I was telling you about. If, say, for example, that interneuron is communicating with another interneuron in the chain, it very well might. This could be a long pyramidal cell. And it goes all the way up, and it's going to synapse in this here, we'll talk about this more tomorrow, is the gray matter of the brain. And the gray matter, we said, functionally does what? Analysis, right? Communication and analysis. So certainly that's where we're gonna find synapses, that's where we're gonna find axon terminals, okay? That's where we're gonna find dendrites and cell bodies of other interneurons or pyramidal cells. And this is just giving you an example of, in your conscious brain, only when you reach that very, very top synapse do you have a perception of someone driving a nail through your shoulder. If that synapse does not happen, you have no idea that that's happening. So that gives you conscious level perception, and then from that, information comes down, synapsing with a motor neuron, telling the motor neuron, hey, this is pain, we need to reflexively get away from it. You're most likely gonna contract your deltoid and move your shoulder away from the stimulus. Again, lightning fast. Are there a gazillion pathways in the nervous system? Yes, that's neuroanatomy. You guys can get to that in grad school. But we're just talking about basic fundamental units of pathways. That's all we're doing here, okay? Okay, all right, so that's all I've got for this. All right, so